Okay, we're gonna have Beth speak, and she can, you know where you want to speak? Do you want to sit? Okay, do you want the microphone? Okay. All right, so welcome Beth. She's gonna tell us a little bit about herself.
Hello, I'm Bethany. As you can tell from my accent, I am not from the US. I'm from England. I live in a town just outside Oxford, about 50 miles from London. Been to a Hao-Fountain conference. This is also the first time I have spoken in front of an audience of this values. So please be gentle with me. I am now 43 years old. I was diagnosed with Hao-Fountain 3 years ago. I am here today with my husband Simon to meet some people with the same condition. Bessie has asked me to speak to you for about 10 minutes. In that time, I will split my talk into 2 sections. My early life as I was growing up and then my adult life. I will conclude with a quick summary that contains advice I would give to those who suffer from Hao-Fountain Syndrome and to the parents. I will be pleased to take questions at the end. You may find it easy to catch me afterwards. I am here for the next 3 days and I will be happy to answer in more detail any questions that you have. Right, let's go back to the very beginning. I was born in 1982. I come from a normal middle class family. At that time, my mum worked for the government, but and marriages. And I was a pilot in my airline. I have a difficult children who was two years old than me. I started to help and to people of what I wanted when I was younger. The pandemic was not the right way for me when I was just a few days old. I'm usually floppy and not as alert as my older sister had been. I was said, but I had difficulty digesting formula. I was sick after most things. Also, in my early months, I was a terrible sleeper and I would wake up crying about every hour during the night. And then be awake for about half an hour and would often vomit in my class. My parents had a stack of bedclothes, warm suits next door to my cot, and I guess their night routine was not easy. I first slept through the night when I was almost 5 years old. I didn't crawl. I shuffled on my bottom and eventually walked when I was about 2 years old. Still, my walking was not good. I would often fall over. My speech was also delayed quite a lot. I started picking up a few words when I was about 3. Even by 5, I was unable to communicate well. My parents took me to lots of doctors and specialists. This was in the mid-80s. And of course, Hao-Fountain was not being discovered. The doctors ruled out all causes for the slow development. If I could summarise their view, it was that I was behind and I measured against all of the expected development milestones. But in the absence of the condition, they could identify, I assume, as just a bit slow, then often said that might catch up at some point. The first diagnosis came when I was taken for a full day's testing at a hospital again, nothing specific. We found out that my clumsiness and a lack of balance called dyspraxia, and a lack of speech was seen as outside the normal limits. There was also suggestions that I could be on the autistic spectrum, but it was difficult to identify that at such a young age. I used to use Makaton sign language for a bit until I could communicate and talk. It was agreed that I would struggle in a mainstream school. There were a few places at a special school, but the parents found in the main school with a special speech and language unit within it. The trouble was that it's 100 miles away from my home. My parents had a solution and was to move house to next to the school, then wait to secure a place. I started at the speech and language and then I was 5 and my sister also joined the mainstream part of the school as well. This journey was a happy time for me. There were 2 specialist teachers and I had a weekly session on speech therapy. I was in touch with my teacher from the unit. My speech improved slowly, as did my balance. But I had a number of random symptoms that I had some food allergies, I had asthma, eczema quite badly, and joint would overextend and sometimes dislocate. I still fell over and vomited a lot, although my sleeping got better. It was not good. And the big point my parents would make is that I began to show a lot of autistic syndrome, and I couldn't really body language and see autistic symptoms. Thinking I couldn't read other people's points of view. I really wanted to have friends, especially a best friend, but my difficulty is understanding and interacting with people made it really hard to make friends. This was what I found hardest in the early years and in my teenage years. I found this very hard to make friends. So let me now move on to my secondary school years. I changed at 11. At age 11, I certainly improved and was decided that I would go to mainstream secondary school, but then I would need some additional help. We moved house and I went to a local school, and I was a teaching assistant for a few hours a week. Looking back, my time from 11 through 19 was very challenging. To others, I didn't look as I had a problem, but my difficulties interacting with people made it hard for me to make friends, so I walked along. I would also get into trouble at school, not because I was naughty, but because I was trying hard to navigate complex while being a teenager. I could just about cope with schoolwork, but it was clear that I wouldn't get many school exam qualifications. But I managed to come up with 3 GCSEs. My parents spent a lot of time helping me with work, and I bought them all the way and found lots of ways to avoid doing it. I didn't know how my mum and dad put up with me. I realised that I had problems and I wanted someone to fix them. Of course, that was not going to happen. I had lots of medical conditions and seemed to spend a lot of time at the doctors, but looking back to my teenage years were okay. My slow development continued and I didn't start my periods until I was 16. They were also painful and not regular. And I went on to the concept until I was 18 to try to make them better. That worked well. I stayed on the medication for about 15 years. I left school at 16. I went on to a vocational officer course at college for about 2 years. I learned a number of skills that would help me to find a job. And I quite liked the call from again when I struggled to form friendships. When I was 18, I did so many other things at the local level place where my dad used to work. I worked there in the movie graphics department doing a photocopying job, making booklets of such like. I really enjoyed that. The next year, I found a full-time doing the same sort of work, and I did a couple of other jobs, and then ended up back on the amazing Air Force Base. And a few years later doing the same job. I really enjoyed this job. My life had changed a lot. I passed my driving test and I bought my first car. My parents separated and I found that very unsettling and I found it hard too. They took a loan out and bought me a small house and I learned to live independently. The most scientific change was I met my lovely husband Simon. We moved in together and then we got married. And when I was 29, we've been living together for 30, got married. And we've been married for 30 years. We do a lot together. I like swimming and also Simon's a very good scuba diver, so he taught me to dive as well. We travelled to many countries and have dived in some of them. We went to New Zealand for our honeymoon and and then we were disappointed was that we were unable to have children. When I did not get pregnant, I went to the doctor when they found out I'd gone through an early menopause. And I did 3 months IVF, but then it didn't work. So it was during this time I had genetic testing showing that didn't tell me to be diagnosed with Hao-Fountain and it is thought that my gynecological problems are part of that. So we are now just up to date. Let me summarise my views on my life so far. I've always lived with Hao-Fountain and I've spent my first 40 years knowing that there's something wrong with me, but knowing what. I also felt that it's unfair that I have a lot of medical issues that most people don't have. And of course, it is unfair, but now I know that there is a single cause to these problems, and it's just that love that I have Hao-Fountain. I have learned that it affects different people in different ways. And so, to all those parents out there, we must just assume that I am just typical. But then again, perhaps I am. Life has been more difficult for me for most people, but also that I am, in many ways, I've been so lucky. My mum, my dad did so much for me, when I was little, and for do today. And their years of work would allow me to live independently, to own my own house, to get a job, to drive myself to and from work. And I'm so lucky to have found Simon, who is the love of my life. We do so much together, and of course he is here with me today. And let me finish with the advice to those children with Hao-Fountain. Yes, it is unfair, but it could be so much worse. You can still do so much. It requires the work, but it is worth it and it pays off. You can achieve so much, and to those parents, yes, it is unfair, but you can make such a difference. Just look at me. So keep helping your children and don't allow them to distract you from the task. Thank you again for listening to me. If anybody has any questions, I'll try to answer them.
I got a question. How many times have you been skydiving?
I've done it 3 times.
First, I just wanna say, I love that you mentioned that you love swimming and you love travel, because that sounds just like our daughter. But I wanted to ask, what would be your advice for middle school and high school? Because she's currently in middle school. She's a 6th grader, and we're just now starting to see the issues with her learning disability.
All I can suggest is just stay positive and just help her. It is a hard move, but just stay positive and just say it'll be fine.
Thanks so much for your story.
of hearing that. You know, we talk a lot about challenges or deficits or things like that. What are some things that you feel like you were especially good at? You know, whether that's, you know, related to this or you know, what would you feel have been some of your successes?
Probably, I like swimming, probably, I've got no fear. I love jumping out there, traffic. Oh, yeah, three, tandem parachute jumps and one, static line jump.
Did you ever remember experiencing a lot of sensory issues growing up?
Not really.
Okay, thanks for being so honest about the social part of this.
Right.
Wonder if you could talk about that a little more. You know, you said it was difficult to make friends when you're younger. Did that get better over time?
I think a bit of like mixed emotions really. I've got some quite good friends I stay in contact with, but I think it's harder for people to like reading with people and understanding what you want to say to people, because they might misjudge the way you talk and think there's something wrong with her, but we don't know what. How did you met? How did I happen? We were out one night in Oxford in a nightclub. And I got pretty nice one night. And he was standing at the bottom of the stairs. And the back at the bottom said to him, I'll let you catch this one. So I threw myself downstairs.
So, you and I have spoken a lot yesterday. Tell me about the other adventure things that you do, like, you said you were an adrenaline junkie, and you showed me, do you remember yesterday, you showed me a video of what you did?
Yeah, so, there's a place down in Cornwall in the UK, on the south coast, called the Eden Project. I don't know if any people know that. So they've got like a zip wire, and both me and my husband have done a zip wire over the top of the orbs in the of the orbs. And basically, the thing for plastic. It was a high thermal in the UK.
But this thing wasn't, okay, so. A lot of people sit on a zip wire or hold on going around. They both did it like Superman. Like, so, many props to you. It's.
And they've traveled extensively. I mean, obviously New Zealand, but many places in the US as well.
Is there anything in particular your husband does to help support you or be there for you that stands out?
Yeah. He did quite a lot with me.
Money.
I wanted to just say that I'm impressed that you have so much memory, like your mom has so many memories of you as a baby. Cause you really spoke to me and talking about the challenges that your parents had with you as a little one. I struggled with that with our daughter. And so I really resonated with a lot of that, so I appreciate that, but as a mom, that's hard. And so to know now that like, there was something that caused all the sleepless nights and I just thought it was colic and eating issues and the delays. I appreciate your parents remembering that and sharing that with you to share with us.
That's fine.
And I'll say, even though you say you don't remember any sensory issues, by virtue of the fact that you like skydiving. I what's that? The stimulation, those things.
Do we have any other questions? For Beth? No? I'm gonna,
What do you appreciate most about your boyfriend?
Very hard.
Yeah, you do. So now I'll push back. Thank you. Thank you very much.
okay, so I think we're gonna move on and start with, Sarah and Isabella. I think you're gonna do, okay, let's see. I'm gonna just unhook here.
Do I have a little less time to stay on schedule or?
So it's a section skip. Yeah, it's all good. I mean, we're running late, but I think we're gonna be good.
Okay. Just making sure.
No, I'm gonna leave it up because we're gonna hook back into David eventually.
Sorry.
Hello. Hi, everyone. My name is Isabella. I am a senior graduate student, PhD candidate in cerebrologist lab at the Dana-Farber and at the Harvard Medical School. I'm really excited to be here. If you're wondering why I don't have a Boston accent, it's because I grew up in Kansas City, but I'm gonna tell you about a project that recently was accepted for publication, so hopefully you'll be seeing it out by the end of this year. So when you first look up USB 7 online, the first thing that you see is that it's a deubiquinase. And what does that mean? So to take a step back, this is part of the ubiquitin system in the cell. So in the system, we have ubiquitin, which is a protein tag that marks proteins for a variety of different pathways and roles in the cell. So imagine that you're moving apartments and you're using those colored tags, tell the movers, this goes to the kitchen and this goes to the upstairs. That's kind of what ubiquitin is. And deubiquinases or DUBs, these are the proteins that are involved with removing ubiquitin or taking that tag off. And this is super important. Like if you mislabel something, you don't want it going to the wrong place. And it controls a lot of different pathways in eukaryotic or human cells. And as you can see with a variety of downstream things, you can control protein-protein interaction, localization, or abundance. There are roughly 100 human deubiquinases or DUBs identified. The largest family are the USPs, the ubiquitin-specific proteases. So of that is USP7, which is the star of today's presentations. So I'm going to talk a little bit, a quick overview of the structure of USP7, as it's going to be relevant a little bit later in the talk. But so USP7 has 7 domains. So the first one is a track map domain. So this is important for recognizing substrates. It has the catalytic domain, which is in the dark purple. So this is where all the catalytic function of the enzyme. So where the scissors are for removing ubiquitin from targets. It has 5 consecutive ubiquitin-like domains that help kind of orient the protein and its substrates. And it has, more importantly, a C-terminal tail. So it's the last little bit there, 19 residues that kind of loops back and as you can see, it's kind of hooking back to the calic domain. That's super important for the activity of the protein. So USP7 uses the C-terminal tail to activate the protein, and it does that specifically. So it's a cysteine protease. It uses a catalytic cysteine, and that's one of the amino acids that it's using for its scissors. And when it's coordinated with its substrates and the C-terminal tail, you have your catalytic cysteine now in a productive conformation striad. A fancy way of saying that the chemistry aligns for it to cut ubiquitin off of things. So we were wondering in our lab, we had previously done a lot of studies with deubiquinases. We focus on developing small molecule probes or small molecule compounds that we can use to study deubiquinases. And we were wondering if we could take advantage of this mechanism to find something that could maybe mimic the C-terminal tail or activate the protein in the same way. So previously we had run a high throughput screen with the Novartis Fast Labs. This was work from a previous graduate student, Anthony Barca. And he used this assay, which is a pretty standard assay for initially finding or measuring ductility, where you take the ubiquitin and you attach it to a fluorophore. In this case, it's a rhodamine 110 probe. And when you have ductility and you're cleaving the fluorophore, you get fluorescence. So it's a readout of activity. How active is your protein? So if we're looking for inhibitors, we want to see no fluorescence. But in this case, we were looking for an activator. So we're going to be looking for increased fluorescence. So we took that data set and we reanalyzed it, now looking for things that were increasing USP7 fluorescence. And we came across this compound, MS8, which we resynthesized in our lab. And the first thing we noticed is that it seemed to be increasing the fluorescence of USP7 alone, and it wasn't increasing the fluorescence of the other deubiquinases, which started to hint to us that this wasn't just autofluorescence from the compound. But of course, you still need to do a lot of other experiments to confirm that this is the case. And it wasn't just some fluke in the assay. So one of the first things we always check in our lab is that it's selective and that it's increasing the velocity or the initial velocity of the enzyme, so the actual activity. Selectivity is super important, because as I mentioned, there's 100 other deubiquinases, and there's more than half of them are these USPs. They have very similar folding patterns, and they're all cysteine proteases. So, or 6 of the subfamilies are cysteine proteases, right? So you don't want to have compounds that are hitting multiple doves, because then anything you're testing in cells could be potentially other doves having an effect as well in the cell. So you really want to make sure you're using selective probes. And we see that our compound is indeed very selective for USP7. It's the only enzyme that's being activated. And this includes when you compare to USP47, which is its closest homolog. So next we wanted to check in a non-fluorescent based assay that this wasn't just an artifact. So for an initial check, we use this diubiquitin. So now instead of cleaving ubiquitin from a fluorophore, we're cleaving 2 ubiquitins into monoubiquitin. And we can see in the top one, so in K48 linkages, we see activation, and with K63. So our compound is able to activate USP7's cleavage of 2 ubiquitins into 1. And it doesn't do this with linear ubiquitin. And that's actually in literature, USP7 does not recognize linear ubiquitin. So we're showing that our enzyme is being activated for endogenous substrates, and we're not just activating general ubiquinase activity. As a second orthogonal assay, we used a ubiquitin covalent probe labeling experiment. So for this, we're now similar to how we did with our previous experiment. This was an assay that was developed for inhibitors. So typically we're looking for blockage of this probe. Ubiquitin is about just under 10 kilodaltons. So you can measure its covalent labeling to deubiquinases as a mass shift on a gel. So that's why you see the lower band is going to be unlabeled protein, and the higher band is going to be labeled with ubiquitin. So with our activator, rather than the blockage of the labeling, we're looking for increased rates of labeling. So going from left to right, the first plane doesn't have, well, you have your ladder, then you have your first plane that doesn't have any probes, so we see just the bottom band. Then we have no compound, but we have our ubiquitin probe, so we see some labeling after 2 hours. With our compound, we have significant increase in labeling. Another thing that, if we have some time, I'll talk a little bit more about is we did a small medicinal chemistry campaign in our lab where we were changing different features to the compound to see if we can improve the potency of the compound. So one of the first things we did was flip that oxygen in the furan ring. And we found a compound that, although it has just about every chemical feature the same, changing that one oxygen completely removes any ability to activate the protein. So this helps us gain a little bit more confidence in the structure of it being genuine for an enzyme, because enzymes are very particular where you put things near them. And then this is just showing the ubiquinorodamine, the diubiquitin cleavage, and the pro-labeling, same through experiments I just showed, but with our negative control, and we don't see any change. We also see this with the chiral centers. So MS8, our compound, has a chiral carbon, so it has two different conformations in 3-dimension space. And if you separate them out, we can see that one is preferred over the other. Enzymes are 3-dimensional, so USP7 will prefer, if it's a genuine interaction, we would assume that the chiral center will have an impact, and it does. This is something I would like to also point out for XL177A, which was an inhibitor developed in our lab. We do have a chiral separated compound, XL177B. That's a great negative control because it's the exact same compound, just one chiral center separated. So now that we're pretty sure this is a real activator for our enzyme, we wanted to start figuring out the mechanism and where it was binding. So the first thing we did, so this is work done by a postdoc who was previously in the lab, Shirafi. She took the USP7 catalytic domain and decided to take our compound and pose it computationally on a computer in different positions. So there are two really well characterized pockets on USP7 on the catalytic domain. One's the distal pocket, which is in green. That's where the Genentech GME compound binds. And then you have the S3S5, which is also the catalytic channel. So that's just where the scissor machinery is. And that's where we have most of the inhibitors right now. So that's XL177A, you have a couple others with a similar scaffold. And more recently, this wrapped USP7, which is really great if you're trying to decide what probe to use in cells. But then there's also, as I mentioned earlier, that C-terminal peptide binding pocket, the CPB, which is where the C-terminal tail of the full-length protein comes and wraps around. So we put our compound in all those pockets, and then we ran a simulation where the protein moved through the computer, and we see that our compound is really stable in that C-terminal binding pocket and not the other two. And actually something really cool that I have a movie here. So wiggling there, that's USP7's catalytic domain, and then in green, that's our compound. So in the simulation, it moves around, and it's kind of finding these non-specific interactions. Then after a bit, right there, it lands in the C-terminal peptide pocket, and it stays there for the entire rest of the simulation. And she repeated this 3 times for every single pocket, and in every case, it would pop out of the other 2 pockets and find its way to this one. So, but of course, we can't just trust a computer, we have to do it again in other ways. So this is where we started collaborating with Nova Lab, largest shift compound were in fact belonging to that same pocket that we identified computationally. So the more painful, the larger shift that we see. And if we just zoom in on that interaction, this is just an example of what our data looks like. And then biochemically, we went back and mutated some of these key residues in the pocket, made sure that those mutations did not impact or lose activity. But with our activator, we can no longer activate now. So this is now biophysical and molecular confirmation that our compound is binding this pocket. So of course, that begs the next question, right? So we have a compound that's activating and it's binding that same pocket. Is it mimicking the same mechanism that USP7 has in full length to activate? So the first thing we did was superimpose the 2 things. So this is our compound in that pocket, and then in orange is the tail that normally is in that pocket. Right off the bat, we can see a lot of similarities chemically. So the key lysine residue of the tail superimposes with our dimethylamine of our compound. So very similar chemical parallels between the two. And we have a key hydrogen bond that's kind of hard to see in this angle, but our compound has a carbonyl that hydrogen bonds to the protein in our predicted model that the tail also does. So next, we set up this competition assay where we fluorescently label just the C-terminal tails of USP7. And we wanted to see, using fluorescence polarization, if we could compete off the tail. And by the downward trend, we do see that our compound is competing off the tail. This also, we can see if we were to take the full length protein, which gets activated, but not to the same extent of just the calyptamine, but we take out the C-terminal tail, we now get the full extent of activation that we were seeing. So it's really confirmed with us that the two are competing for binding in that allosteric pocket. The next thing of trying to work out the mechanism of this compound was seeing if we see any changes in the affinity for ubiquitin. So this is similar to the previous experiment, but this time we have fluorescently labeled ubiquitin. And we see that our compound is not changing the affinity of ubiquitin, which is matching literature for the C-terminal tail. And then using again NMR perturbation shift data from the Vesnov lab, we can see that our protein with the ubiquitin already loaded, so already in an active state, has a much tighter affinity for our compound. Which is again matching that stepwise sequence that is reported in literature for the kinetics of the C-terminal tail. And this is something we saw also in our modeling. So the dark black line now is the simulation, but now with ubiquitin present, and we can see very striking stabilization of the compound with ubiquitin present. All right, so summarizing mechanistically, what we found so far is that we have figured out where the compound is binding in this allosteric pocket of the calyptamine of USB7. We know that it's stabilized by the active state of USB7. And we know that it competes with the C-terminal tail. So that kind of drives us to the next question. Can we activate USB7 in a cellular context? Does our compound even enter a cell? It's something you always want to be thinking about with small molecules. And is it engaging the target? There are a lot of other things in the cell that could be precluding your compound. And then that also makes us think, can we activate Hao-Fountain variants? Especially in the case of patients who have truncations after the calyptamine, where they lose the C-terminal domains. Those C-terminal domains are crucial for activity. So can we use our compound to mimic what the enzyme is missing? That second part of the question will be answered in the next talk, so I'll leave you guys in suspense for now. But for the first part, I'll talk about the assay that we use for measuring cellular USB7. So this was a really great assay developed by the Harris lab at, I believe, UT Health San Antonio. So it is a GFP reporter assay. So essentially they have GFP, which is green fluorescent protein, so it has a green signal, and they modified it so it's constantly ubiquitinated and constantly degraded by the proteasome. But then when you introduce a deubiquinase, you start to have the rescue and then you get a green fluorescent signal. So we can measure USB7's activity in a cellular context using this. But as we mentioned before, USB7 full length is extremely active and we weren't even sure if our compound would be able to activate since the C-terminal tail was present. So to do the studies, just to make sure that our compound enters the cell, we used a transfected version of our truncated protein. So this is USB7 with the delta tail mutation. So this was just checking that our assay worked and that we're seeing loss of function with that delta tail mutation, which we indeed saw. So our USB7 full length is pretty active, and then our the loss of just those last bits of the tail, you lose just about all activity. And then when we introduce our compound, we see really nice activation with it. This confirmed with us that our compound does work in a cellular context and can find USB7. We also, I didn't mention it with the previous cell line, but we always check that our compounds are not inducing cellular toxicity. It's one of the first things any cell lab should do when you're testing new compounds, because some compounds could have toxicity in very distinct scenarios. But so we have no toxicity, even up to 72 hours with compound treatments, up to 200 micromolar, which is really nice. And then we see when we transfect in that delta tail mutant into a cell line that does not have any USB7, we can see changes in downstream substrates, P21, which is downstream of P53. And then we confirmed this again, so we took lysates that were treated with our compound, and we ran that covalent probe labeling experiment. We do see increased probe labeling with the activator treated cells. So I had a section on chemistry, but I feel like I might just skip over that section for now. But just as a big summary, we've been working on different parts of the compound that can improve the potency and hopefully find something that's better at activating. So yeah, thank you. With that, I would like to thank my lab in particular. Andres is a postdoc in the lab who did a lot of the cell work and got that assay from the Harris lab running. Mona, who was a previous postdoc, did a lot of the computer simulations and the synthesis for the activator. Of course, my collaborators who are here, also thank them and all the funding. Thank you for listening.
Is Emily's like complimentary to you? Okay, so do we want to just have Emily just come on up since your presentation is kind of our complimentary? Yeah. Are you on the same thing?
Yeah.
Yeah. Oh, is yours on mic?
No, it's on
No, I have it.
Okay, I was like, okay, do I need to? Okay.
Just let me close mine to make sure. It's the- Okay.
It takes a second sometimes. Because I kept turning it on and off.
Let's see, it's in Puerto Rico. Also. Yeah, no, I think I have to click enable editing to get into presentation mode. Yeah. And then the arrows.
Okay. Hi everyone, my name is Emily Korchak. I work for Dr. Betsinova over there, at UConn Health, so in Connecticut. We've collaborated a lot with Dr. Burlack and Isabella, so this is kind of like a follow-up story that goes with their, presentation of MS8. So this first part of the talk is gonna be about, a paper that just got accepted. We got an email about an hour ago. It'll come out next week. So look out for that. But the second part is gonna be some unpublished data that I've never really shown anyone, so hopefully you can follow along. I've gotten some great introductions from everyone, so I feel like it's very useless for me to introduce Hao-Fountain Syndrome. As you know, it's caused by mutation or deletions in USP7. USP7 is a deubiquitinating enzyme that removes ubiquitin off of substrates and therefore changes their cellular fate. So USP7 is really critical as a protein, as it's involved in numerous pathways, including DNA damage, the immune response, apoptosis, and just many more different pathways. But one specific pathway that was introduced by Dr. Schaff earlier today from the original Hao-Fountain Syndrome paper in 2015 is the WASH pathway in endosomal protein recycling. So here, USP7 forms a tricomplex with MAGEL2 and TRIM27, where TRIM27 will ubiquitinate the WASH complex, which enhances endosomal actin and protein recycling. And at the same time, it can auto-ubiquitinate itself and target itself for degradation. So USP7 is kind of balancing this out by deubiquitinating the WASH complex, which regulates the levels of WASH activation, but it also deubiquitinates TRIM27 and rescues it from being degraded. So it's really critical. And so they had shown previously that mutations and deletions in MAGEL2 lead to Hao-Fountain syndromes and Prader-Willi syndromes, but they then discovered that mutations in USP7 also have similar phenotypes with Hao-Fountain syndrome. And so when USP7 doesn't function properly, TRIM27 just keeps auto-ubiquitinating itself and targeting itself for degradation, which leads to a decreased endosomal protein recycling. So getting into Hao-Fountain syndrome, when I was writing my thesis, I kind of wanted to come up with all the mutations I could find online from ClipVar, from Dr. Schaff's lab,
from all the publications. So here is a compiled list of all the missense mutations on the top and truncating mutations at the bottom, not including deletions of genes. But USP7, as Isabella mentioned, is a 7-domain protein with the TRAF and terminal domain, followed by the critical catalytic domain and 5 ubiquitin-like domains. So the catalytic domain is what I focused most of my thesis on. When I first started 5 years ago, I was looking at 11 mutations in the catalytic domain that were the first 11 mutations we were given. So they're highlighted in blue here, so that's going to be the first part of my talk. And then I'll go outside of the catalytic domain and touch upon substrate binding domains. Okay, so those 11 missense mutations in the catalytic domain that I've studied are shown here on the structure of the catalytic domain, which Isabella kind of started introducing. So here, as you can see, the catalytic domain takes the shape of a right hand, and there's at the top the ubiquitin binding fingers. And then you've got the catalytic cysteine, which is critical for this cutting of the ubiquitin. And you can see that all 11 mutations are kind of scattered throughout the entire domain. So they could have different effects on the protein. So our major hypothesis was that mutations in this domain could affect the stability of the protein, so its ability to fold, substrate binding, so its ability to bind to ubiquitin, or the activity of the enzyme. So we started by looking at the structural integrity of the enzyme, where we did a thermostability assay looking at intrinsic tryptophan fluorescence. So basically, easily this just tells you that the melting temperature can speak upon the integrity of the protein. So if we compare the wild type shown in black to all of these 11 mutants in the different rainbow colors, we can see that most mutants have similar melting temperatures to the wild type, meaning they're most likely properly folded. However, when you come into the reddish, pinkish colors, you can see that there's a decrease in melting temperatures. There's one of them we couldn't even get a melting temperature, which means they might have some sort of defects in folding of the catalytic domain. So throughout the rest of my talk, these colors are going to be followed for all mutations, with bluer colors being more stable and reddish, pinkish colors being more unstable. So most of them were stable, so we decided to look then at substrate binding. So as I mentioned, the catalytic domain binds to ubiquitin. Ubiquitin here is shown in gray as this surface-like structure, and you can see that it binds to the fingers. So we would expect that if you have a mutation on the fingers, there could be some sort of defect in ubiquitin binding. So I'm pointing out one specific mutation here, E345K. So we're changing a glutamic acid to a lysine in terms of amino acid changes, and that's a change in charge. So we would expect to have some sort of inability to properly bind to ubiquitin. So we proceeded with doing a nuclear magnetic resonance spectroscopy, which in a super easy manner, you're able to just look at two proteins binding to one another. So if you get this beautiful spectrum full of these peaks, is what we call, each peak is going to be a specific residue in your protein. And if you add increasing concentrations of ubiquitin into your protein, you can see these movements, which are called chemical shift perturbations, going from red to magenta. And that means that there's binding between USP7 and ubiquitin. So that's for the wild type. But we did that for all 11 mutants, and here I'm just showing one of them. Where we see that there's no changes in these chemical shift perturbations, or very little. So there's clearly a defect in its ability to bind to ubiquitin. And what's great about NMR is that you can then quantify this data and get binding affinities. And so I did that for all 11 of the mutations. And you can see that most of them have very similar binding affinities to the wild type. Maybe some are a little tighter, some are a little weaker, but E345K clearly is the outlier with severe defects in its ability to bind ubiquitin. So the last step of this project was to look at the activity of the enzyme. So right now, I'm focusing on the catalytic domain alone. And what we're able to determine for the activity is the ability for the protein to deubiquitinate substrates. So we have this small fluorogenic substrate that we're able to track over time for its fluorescence, and then you can get kinetic parameters. Here I'm showing kcat, which just speaks to how well your protein functions. And so what was interesting with this, we saw two trends. On the right-hand side, we see a loss of function, so less activity for these proteins. If you look at the colors of these mutations, they are pinker for L416-9R, B45G, K420E. So those are unstable mutants. If your protein's not able to fold properly, it makes sense that you won't be able to have a good activity. E345K, as I just mentioned, is deficient in ubiquitin binding. Even though it's very stable, if it can't bind ubiquitin, it's not going to function properly. But what was really interesting and kind of makes us really interested in the duplications that were mentioned today, is we saw some hyperactivation for some of the mutations. And so we don't really know why we saw this hyperactivation. It's very unexpected. We have some suspicions for certain, for example, G392D. We did another study that's also published in PNAS, where we saw that the fingers of the binding domain for ubiquitin are more dynamic, so that could speak to that. But this was just for the catalytic domain alone, and what patients have is the full-length enzyme, so it's kind of important to look at it in that context. So we did the exact same assay now with those same mutations, but now in the context of the full-length enzyme, so not just the catalytic domain. And we see that it is indeed a loss of function for all mutants. So even though the trend remains the same with the bluer hues being more active than the redder ones, we still see that they're all loss of function. But to tie this up with Isabella's talk, how can we actually help? If these are all loss of function in the full-length enzyme, can we actually rescue the activity of these variants? So we ran the exact same assay I just talked about, where we're looking at the activity of the protein, but this time we just added our small molecule. And if you compare the activity, we can see that for the two first bars, we can see that the wild type can be nicely activated. And we can see that the mutations that are stable and that showed some levels of activity can be increased in activity. However, if the protein is unfolded or has severe defects in ubiquitin binding, it doesn't seem like we're able to rescue that activity. So she explained how this worked, where the MS8 is able to mimic the C-terminal tail because USP7, it has its own autoregulatory activation. So that's critical. So she started bringing this up where we thought that if patients have mutations that create truncations past the intact catalytic domain, so if MS8 is still able to bind, but there's no C-terminal tail, this MS8 would really be promising for patients of that kind. So we went back to our list of mutations and we found the shortest truncation past an intact catalytic domain. E547 star, shown here, is the one that has all the UPL domains missing, but has an intact catalytic domain. So we created that mutation in vitro and we added the small molecule, and we can see that we can actually really nicely rescue this mutation. I think from its original activity, we're able to rescue it 47-fold, which is really exciting. So this would really represent kind of all truncations past that mutation and provide hope for all those lacking the C-terminal tail. And so Andres in Dr. Burlak's lab also was able to bring this into cell work to kind of see how that works. So Isabella introduced this GFP construct that they're able to use in cells, which is what they used here. And we can see that with increasing concentrations of this MS8, we're able to nicely activate this mutant, which is very promising for the use of the small molecule later. Okay, so that was for the published work that's coming out next week, but the other domains are also very critical and important to study. So what we started doing is looking at substrate binding domains. So TRAP and UBL1/2 are substrate binding domains, and you can see that they have their own set of mutations. So we hypothesized that if there are mutations in those domains, they may affect substrate binding. So From the original Hao-Fountain Syndrome paper, they showed that MAGEL2 interacts with USP7 between its residues 820 and 1034. From prior knowledge of how TRAF and UBL12 interact with substrates, we know that UBL12 recognizes a lysine-rich motif, which there's only one motif in that sequence, while TRAF recognizes the PXXS motif, shown in blue here, where there are 7. So we kind of went on tried to see which of those motifs binds the tightest to TRAF and kind of confirm this interaction. So we created peptides containing those motifs. So starting with UBL12, there was only one peptide, and we were able to do NMR, where we just look at the binding of this peptide to the UBL12 domains. And I'm showing the structure of UBL12 here, with the mapping of NMR. So if it's red, that's where we expect the peptide to be binding. And using AlphaFold, I was able to model in those peptides. Well, this one peptide, sorry, there are 5 because those are 5 AlphaFold models that were created, but it kind of matches nicely with the NMR data that I got. So we found an affinity of 64 micromolar. And then for the TRAF domain, we used all 7 of the peptides that we created, and we found that there was a clear winner from residue 956 to 968 with 37 micromolar. And we did the same on this domain where we put AlphaFold, and we can see that it where it's supposed to bind, and it matches all other published TRAF binding peptides as well. So there's really this bipartite binding between UBL12 and TRAF together that are able to bind to MAGEL2. So looking at specific mutations in TRAF, here I have 3 that we have worked with so far: F118L, H11Q, H111Q, and R152C. So if you look at where those mutations are located, you can see that F118L is exactly where this peptide is binding. So we hypothesized that this would really have some sort of defect. And so we did NMR again, and looked at the affinity of these mutants and compared it to the wild type for this one specific MAGEL2 peptide binding to TRAF. And we can see that H111Q and R152C have similar binding affinities to the wild type, but F118L has severe defects in the binding. And we used an orthogonal technique called ITC, which you can just also get binding affinities looking at interactions. And we can see that we also have some severe defects for this mutation, so we really confirmed our findings. As for UBL12, we studied 2 different variants, I766T and L757P, and those were unstable, so I wasn't able to get any affinity data for that, so there might be some sort of defect in folding of those domains. So that would be just something to look into the full length as well. So with that, I've shown that Hao-Fountain syndrome mutations can affect the stability, activity of the enzyme, and substrate binding. Structurally stable variants of the catalytic domain that are not affecting ubiquitin binding can be rescued with a small molecule, at least in vitro. But we've shown that we can also bring it in vivo with truncation mutations, which is really like the take-home message, which is promising, in my opinion, that this MS8 mimicking the C-tail could really provide hope for truncating mutations past the catalytic domain. So this was just the first stepping stone for potential future therapeutic applications, and just the first understanding the effects of these mutations basically on the USP7 protein. So with that, I'd like to thank the UConn Health graduate program and our department, Dr. Betsinova and Dr. Korshnet's labs, as well as our collaborators, Dr. Berlag and Isabella specifically, Alison O'Neill, which is also here, and Dr. Christian Chen, and our funding, which we got funding from this foundation. So thank you very much, and I'll take any questions if you have any.
But just from a layperson's perspective, so as these are possible therapeutics that have been determined, it looks like on the computer model in just chemistry, with the proteins, but then also in the cell itself, in terms of just single cells in this case, right? So with the next step, be looking for multicellular organism models that.
Yeah, so there's definitely a lot of steps
Yeah,
coming. Yeah, there'll have to be like a mouse model, all of that,
Yeah.
which we don't know if that'll work. But I'm not at all into cell work, so I don't know much about that. I think maybe Isabella knows a lot more. No, never mind. But I don't do cell work, but there's a lot of steps before this could be brought into clinical trials and to become something. But it is exciting that we're at least able to see this activation in cells, that this small molecule is able to go through the barriers, go into the cells and activate.
If I can maybe just, interject too, right? And another thing to keep in mind is these are, these aren't neurons, right? These are cells where we've engineered, USP7 that has patient relevant mutations. So, right, Allison O'Neill is now developing the system to at least look still in a cellular context is the next logical step. What do they do there? And then, ultimately, hopefully get to, animal models, maybe organoids along the way. But, truth is, these compounds need derivative optimization, which are really ready for an animal model. But you bring up such an important point about the need for more and more models.
Yeah. Thank you.
You're able to discern, for example, with the specific mutation that it has severe binding defects. Have you looked at any of the phenotype data for that mutation? If not, are there plans to do so?
I don't have a lot of data on phenotypes. So if we get provided the phenotypes, I think we could. There's some that we do know that coincide with the mutations I've studied, but I haven't personally really linked them together. But in the 2024 paper from Dr. Schaff's lab, they came up with this severity score, which I think would be great if all families would participate in being able to create a severity score for each mutant, and then putting together the severity score and then what I see in vitro, and if we can find correlations between substrate binding defects, activity defects, and the severity score. So I think that would be really, really cool. But so far, there's only, I think, 3 or 4 mutations that we could put together, which is way too little to make any conclusion. Okay, thank you.
All right, I got it. Thank you so much. I have to set David up because he's doing his remote. So I just need to do that. Okay, and David, can you hear us?
Yes, I can hear you. Can you hear me?
I'm gonna try to turn it up because I don't know if it's hooked up. Let's see. All right, can you talk again?
Yes, can you hear me? Is this good?
No.
Perhaps you can put the microphone next to the speaker of the, so there was a portable microphone that the speakers were using.
I am just gonna try to see if, I don't know if there's a way to get the speaker to work through the
Okay.
All right, David, can you try again?
Yes, can you hear me?
It's just not coming through the speaker. I did that. Let me see.
Perhaps Was there a portable microphone? Perhaps if you put that next to the speaker of the laptop, then.
So speakers, which one? Same as system? Okay, let's see. There's a couple different options. Do you want to try saying something again?
Yes.
There we go. Okay.
Is it
Thank
better?
you.
Oh, good. Okay, thank you very much. Really excited to have the opportunity to speak to you. And my apologies that I couldn't make it in person. So bear with me, and I'll try to do this remotely. So I wanted to give a very quick overview of a couple of drug repurposing campaigns that we ran. And this is really an introduction to Janelle's talk, who's going to follow me. And I wanted to do this so that you have an understanding of what do these different drug repurposing programs encompass and what is a potential output. But first, this first slide is really an overview of how people go about repurposing drugs. And repurposing drugs is basically finding a new purpose for an existing molecule. So it could be a drug that was originally developed for Parkinson's disease, and you figure out it also works for high cholesterol. There are a couple of ways that people have done drug repurposing, and one is just serendipity. You just do an experiment in animals and you figure out that the molecule you thought was a great anti-anxiety drug is also perfect in losing weight, for instance. An example of this are PD-5 inhibitors. A second way is to really understand the biology of disease, the new disease that you want to address, figure out the molecular pathways, the biochemistry. You can do this also with modern technologies like transcriptomics, proteomics, metabolomics, and nowadays, of course, you use AI as well to figure out if there may be a drug out there that would work on the defect that you've identified, the molecular consequences of typically a mutation in a gene. And then the last approach is basically a brute force approach, where you take an assay, so a cell model or an animal model, typically not a mouse or rodent, but this could be a worm, a fly, or a yeast, and you just screen every compound that may be relevant in this assay to see if it does anything positive. A couple of examples. During the COVID-19 pandemic, people tried this and they ran assays and screened for compounds that would inhibit COVID-19 replication. And people have also done this for rare diseases. For instance, in fly models, screen lots of compounds and find a molecule that potentially works. The approach that we typically use at Charles River is to screen in patient cells. So we don't screen in worms or flies. We use patient cells. And I want to share with you 3 different examples. What these have in common, that they're all very rare diseases, ultra rare diseases. In some cases, there may be only 2 other patients with that same disease in the world. And it's all family foundations, so individual families who funded these programs. And you can see already there are very different diseases. So there's a mitochondrial disorder, there's a DNA repair disorder, and then there's a disorder that we don't actually really understand what the underlying mechanism is, just the gene. So the approach that we typically take is to screen as many compounds as relevant. So these are typically molecules, small molecule drugs that have been approved for another indication or have been in the clinic. So they could be drugs that are still in phase 2, so they have not yet been approved by the FDA or other regulators, but at least we understand what kind of dose you can give to people, if they're safe, well tolerated. And this comes to about 5,000-6,000 molecules. Then the assay that we typically use is patient cell derived, but it can also be cell lines with particular mutations knocked in. These can be skin fibroblasts from patients, or of course also iPSC, so induced pluripotent stem cells, which you can then use to differentiate into neurons, for instance. And what you typically want to use is patient cells and a healthy control. So that can be just a random control, but ideally you'd have this from a sibling or family member so that you keep it as close as possible. And then the readout, of course, is very important. It can be something very direct. If you're trying to upregulate a protein, you can measure that protein in the cells. You can look at downstream targets. So if you understand the biochemical pathway that the protein that's mutated in disease is involved in, you can look for mechanisms that bypass this protein and affect those downstream targets to different means. Or, and that's the last example that I'll give, it's a completely agnostic approach, which is called cell painting, where you just look at the morphology of the cells, the behavior of the cells, and every assay basically is the same. It's just a different interpretation of what the cells look like. So the first example is a mitochondrial disorder. So this is a program we ran for Kendall. She is now 9 years old, lives in North Carolina, and she has a very rare mitochondrial disorder. So she has a mutation in the mitochondrial DNA. The mitochondrial DNA is tiny. It's only 16,000 nucleotides, but it has a couple of genes that are very important. And mitochondria are like the power plants of the cell. So we use the mitochondria to generate energy. So without mitochondria, everything just goes pear-shaped. She has a very rare mutation. There are only 2 other patients known in the world with this mutation. And the syndrome, Lee syndrome, is really characterized by lots of things going wrong. So neuronal metabolism, immunity. So yeah, she is very poor. What is very special about her mutation is that not every copy of her mitochondrial DNA is mutated. And this is probably the reason that she's still alive, because if every copy of her mitochondrial DNA was mutated, the mitochondria would be dysfunctional. But about 20% of her mitochondria have a normal copy of the gene, and that just keeps her alive. But of course, she is not in a very good condition. So this is called heteroplasmy. So it's very specific to mitochondria. It's a bit like mosaicism for the genes that are in our nuclear genome. So the program is summarized on this slide. So we took receipt of Kendall's cells, her skin fibroblast in this case, and developed an assay, and I'll show the assay on the next slide. Then we ran a pilot screen just to make sure that the assay is robust for high throughput screening. And then we screened about 7,500 compounds, and they are composed of the libraries that I mentioned on this slide. There is some overlap between these libraries, but we just wanted to get as many compounds through this assay as possible. And then after the primary screen, where you screen everything at a single or 2 concentrations, we repeated every hit and did concentration response curve so that you understand the potential dose you need to give to have a potential benefit. So these are just some pictures of Kendall's cells at the bottom, her skin fibroblasts as they grow in culture, and control fibroblasts from an age-matched control on top. And you can see between day 1 and day 3, both cells have continued to grow, so the culture is fuller. These cells have grown to confluency. And these cells are grown in very rich medium. That's how we like to grow cells, lots of glucose. And these cells are indistinguishable in those conditions from control fibroblasts. But the moment you take away this glucose energy source and you give these cells an alternative energy source, galactose, which is a different type of sugar, you can see that the control fibroblasts, they cope well, they just change their biochemistry, and the mitochondria start burning galactose instead of glucose. But Kendall cells, they can't cope with this. So they only survive on glucose. They can't deal with other energy sources. And you can see in the bottom right corner that there are very few cells in the dish. So they really have stopped to proliferate under these galactose conditions. So this is a very stringent assay. It's also a very easy assay to screen compounds. You just grow cells, put them in galactose, and see if any compound allows these cells to proliferate with only galactose as the energy. So we screened about 7,500 compounds and found about 100 hits. And what we did, we not only looked at cells that would grow or have some level of growth, but we also looked at the mitochondria, what the mitochondria look like in candle cells. So that's why we had another set of hits that we combined. We did concentration response curves, and we came up with this list of hits that came through for both cell growth and mitochondrial morphology. I won't go through the identity of the compounds, but what you can see in the mode of action column is that there are some target classes in common that come up again and again and again, like HDAC inhibitors, MAP kinase inhibitors. So there are a couple of protein classes that have clearly been enriched in this assay, probably indicating a biological mechanism by which you can compensate for the mitochondrial defect. So at the moment, we are running some of these compounds in higher organisms like flies, just to figure out which ones are best. Some of these compounds are also going to be difficult to administer because they're cancer drugs. So they're given to patients that have tumors. They're typically not given to children with a chronic condition because of the potential side effects. So that's an area that you always have to be careful. You may get hits and they may be clinically approved, but they could be drugs that are just not very suitable for children or any chronic condition. So now moving on to the second example, and this is a program that we ran for Rian, picture here in the slide. He's a young boy in New York, and he has cocaine syndrome, and this is a DNA repair disorder. But it's a very nasty syndrome because it's like accelerated aging. So his brain is degenerating already very early on, and the life expectancy for cocaine syndrome is very short, typically in your teens. And what Rian's family wants to do is of course identify a potential therapeutic. They're working on two different fronts. One is a gene therapy program that they're running with UMass. And of course, this is a gene replacement strategy. So if this is successful, they can administer the AAV gene therapy to Rian, and hopefully that's once injected and then done. But because it takes time to develop an AAV gene therapy, you need to go through safety testing, etc. They wanted to do a drug repurposing screen because it's run quicker, and potentially you might find something that would sort of tie Rian over until hopefully that gene therapy would be available to him. The assay that we used is a bit different. So we didn't formally use Rian cells. We really used a recombinant cell model. Of course, it was deficient in the gene ERCC8 that is lost in Rian. But what we did was run a reporter assay. So this is a GFP reporter, a green fluorescent protein reporter. And what it does, it captures one of the defects introduced by the loss of this gene in cocaine syndrome. The role of ERCC8 is really to monitor DNA damage as it happens in the genome, and especially in transcribed genes. So anything that is making protein, which is, so DNA is, of course, encodes RNA, and RNA encodes protein. Any gene that is actively being transcribed is monitored extra carefully because, of course, it's important. If it's not doing anything, you don't have to check it every 5 minutes. But if it's a transcribed gene, you need to actively look for DNA damage. Once DNA damage is identified, the cocaine syndrome complex turns down the transcription of that gene so that repair can happen. It's like blocking off a street if there's a pothole so that you can fix the pothole. And then, of course, once the pothole is repaired, you need to unblock the street so that traffic can reoccur. And it's this last aspect that goes wrong in cocaine syndrome. So transcription is shut down, repair in principle can occur, but then transcription is never allowed to happen again. So the gene is shut down. And this also explains the neurodegeneration. Constantly genes are being turned down because there's DNA damage and then they're not turned back on again. So this is the reporter system that we've used. We introduce DNA damage in cells. We allow the reporter that reports the shutdown of expression to come up. Then we wait 24 hours. This is the moment that in control cells, everything is back to normal, but in cocaine syndrome cells, everything is still blocked. And this is the readout for the screen. So you can appreciate that in the images in the top left. So in the bottom right hand of those 8 images, you can see green cells. So this is the reporter 24 hours after the induction of DNA damage. So at this point, there should not be that many green cells, but you can see there are many green cells after the DNA damage induction. And this is the phenotype that we want to reduce in this screen. So yeah, we screen again thousands of compounds. You can see the analysis of the high throughput screen in the bottom right graph. And we picked hits that released the increase in GFP at 24 hours. And here are some examples of those hits. Of course, we did concentration response curves on the left-hand side. And we looked as well at the different classes of these hits. So what kind of targets did we see enriched? And again, we saw a number of target classes enriched, heat shock protein inhibitors, CDK inhibitors, mTOR inhibitors. So clearly a couple of classes were enriched in this case. And hopefully, so together with an academic lab, we're now progressing these hits to further understand how they work and whether they are truly potential therapeutic candidates. The last example is on PAX2. This is an ultra-rare disease. Again, very, very rare. And you can see the family that we ran this project for. And Lena is one of the twin girls that has a mutation in PAX2, as you can see in those sequencing data. So PAX2 is a complex protein. It acts in a complex, but it therefore also has different functions. It has functions in the nucleus, it has function in the cytoplasm. It interacts with all kinds of proteins affecting the endoplasmatic reticulum, mitochondria, ion channels. So clearly a very important protein, but also difficult to sort of capture in a very easy readout, like we've just shown you in the GFP reporter for cocaine syndrome. So we therefore opted to use cell painting as a readout for the drug repurposing screen. And cell painting is a technology that was originally developed at the Broad Institute by Ann Carpenter. And in principle, it is very simple. This is just a way to stain cells as they're grown in cell culture for different compartments of the cell. So you stain RNA, the nucleus, the cytoskeleton, mitochondria, the endoplasmatic reticulum, the plasma membrane. So it's a combination of a number of different stains. And then you use high content imaging. So this is a type of microscopy to really look at all these different stains on a cell by cell level. So in one well, you can have thousands of cells, but you look at every cell in that well with all those different stains. and you come up with a quantitative measure for each of those stains in each of those cells. And then you get a phenotype of those cells, and by comparing a diseased cell, so a patient cell, with a healthy cell, you can see what is different in these cells in the morphology. Is it the nucleus, is it the ER, or is it a combination of multiple aspects? This is how we ran that program. So the beginning is very simple. You seed the cells like you do for any screen. You add compounds, you do the staining, you image the cells, but then the complexity really starts because you need to then run an image analysis. You need to get all those features from those different stains for every cell. You need to process that so that you really have good quality data. You then need to do hit calling based on all those different features. And then what we've included as well is a machine learning algorithm or AI to really understand which of those features are most important in the classification between diseased and healthy, and what are the underlying statistics there. The QC or quality control is of course very complex. It's not only the cells and the images, but it's also all those measurements that are being checked using statistics. Here are some of the data from the high throughput screen. So you can see you actually, once you do all this and you do all this analysis, you do principal component analysis, you can actually get data that looks very similar to the drug reparation screen that I showed earlier. You can clearly see these dots, or every dot is one compound, and you can see that the diseased cells, they are in red, they are really separated from the healthy controls, but there are some compounds that are in between. So those are compounds, hit compounds, that move the phenotype as characterized by all these different dyes from the diseased phenotype towards the healthy phenotype. And you can even do those response curves using that analysis. By eye, it is very difficult to do this. So this is an example where the disease line untreated is on top, the healthy cells are at the bottom, and then the hit is shown in the middle. It's difficult to see, I think, but I hope you can appreciate that the hit looks a little bit more similar to the healthy than to the original disease. But yeah, that's why we use computers. It's much easier to do this analysis. So the conclusions for the cell painting screen for PAX2 are that actually, yeah, this was a very efficient approach. You don't really have to develop an assay because these are the same dyes that everyone uses around the world for cell painting. But of course, the analysis tools are important. So we screened about 6,000 compounds and identified about 70 hits. And then looking at concentration response curves and the targets, we have a number of hits that really look attractive. But of course, again, we need to follow up in other organisms. And the PAX2 Foundation is also making a mouse model of this disease so that these hits can be tested in a mouse model as well. And this brings me to my last slide, and it's really an introduction to the next presentation because Janelle is going to talk about the drug prevention screen that is ongoing for Hao-Fountain Syndrome on USP7. So we started this project about half a year ago, and here we're using an engineered cell line with loss of function of USP7, and of course the isogenic control of that cell line. And looking at the downstream consequences of the loss of USP7 by high content imaging. And of course, we're at the cusp now of screening our drug repurposing libraries. And I just want to mention the team at the bottom of this slide that is responsible to run all these experiments at the Charles River Netherlands site. Happy to answer any questions.
Does anyone have a question for David?
Right now, there's no questions, David. Thank you. Oh, wait, no, we do have a question. Clementina has a question. Yes.
Thank you, David, for giving us an overview of do you hear me?
Can you just make sure?
Thank you for giving us the overview of the work you have done, you're doing. So my question is typically, how you guys decide on an essay and who typically develops an essay before you go to the screen? Which type of essay, for example, how did you decide on self-painting? So yeah, if you can give us an insight into this part of the project.
Yeah, absolutely. So yeah, for PAX2, as I explained, we really didn't understand which biological aspect of PAX2 was going to be most important for disease. It's such a rare disease and it's very poorly studied. That we decided let's give cell painting a shot. Of course, if we wouldn't have been able to differentiate Lena cells from our sister cells, then that we couldn't have screened. But fortunately, we were able to differentiate these two cells where clearly was something different. And that's why we decided to run this. In the case for cocaine syndrome, an academic lab had actually identified that ATF3 response as being very affected in cocaine syndrome, and had built that reporter cell line. So we brought that reporter cell line in. And I think in the case for Kendall, this galactose challenge assay has been used for other mitochondrial diseases. So it's something that we thought was relatively easy to implement, and as you could see, it had a very strong effect. So we typically develop these assays based on what's known about the disease, which phenotypes are which cellular phenotypes are probably translationally most important, what is known about the gene, etc.
Any other questions for David?
Okay, I think that is all, David. Thank you so much for joining us. And I'm gonna let
Thank you.
Janelle hook up. You want to? Yeah. Let me just do this. I don't need to be on. I'm gonna just